In this lecture, we'll briefly explore what are the types of maps that you find in biology or biotechnology. Second, we'll be looking for the basic rules in restriction mapping and how do you arrive at a restriction map given a digestion pattern that is observed on gel. And third, we'll try to reconstruct a restriction map given by the size of bands obtained from a restriction digestion of a DNA molecule by single enzyme and a combination of enzymes. So we'll go through all the intricacies of how a restriction map is prepared. So I'm sure all of you are familiar with Google Maps and all of us have used it at some point of time. So here is my Google map. I start from Intake Housing and come to Red Fort in Delhi. So if you see here, there is a clear distinct path that is shown. And then of course, some of the major features and major towns that are present on the way are also shown here. So you start with Chandigarh, you have Ambala Camp, you have Karnal, you have Panipat, Sonipat, and finally you reach Delhi Red Fort, right? So this is basically a map. So a map is a graphical representation and gives the distance between two features and helps to navigate. And then of course, map can come at several levels and it could be a high resolution map or a low resolution map. So here, if you see, this is a slightly lower resolution map. This is slightly higher resolution, and this is now a very high resolution map where I have a very short distance. It is marked in with all the features that are present in that particular area. And as you use the Google map for navigation, you would see that it is such fine resolution that it shows you where is the next turn and how far it is and which turn you have to take, right? So this is basically your resolution of the map, depending on how accurately, how frequently, and in how many details the features are part of the map. The map may be a low resolution map or it may be a high resolution map. You want a high resolution map would have the maximum details. A low resolution map will have less features, but it will still give you some sort of a reference to follow as you navigate to your final destination. So before moving forward, let's have a recap on restriction enzymes, which was our lecture 1, module 1. And I'm sure you remember that restriction enzymes are produced by bacteria. And they are basically a part of innate immunity to bacteria against bacteriophages. So as soon as a bacteriophage invades the bacteria, these restriction enzymes go and digest its DNA and thus thwarting the invasion. These endonucleases are sequence specific. That is, only when a specific recognition sequence or a certain order of nucleotides is present in the DNA, invading DNA, do they actually digest that DNA. And then, of course, I'm sure you also remember that conventionally, we have divided restriction enzymes into 3 classes, of which class 2 is the most commonly used class in biotechnology. And this is because class 2 restriction enzymes cut within their recognition sequence. And the type of cut that can be produced could be of 2 types. You could have a staggered cut where the top strand and the bottom strand are cut at different positions, or you could have a blunt end cut where both the top strand of DNA and the bottom strand of DNA are cut at the same position, right? So here is an example of HAE3, which is a blunt end cutter, and the remaining BAMH1, EcoR1, and NOT1 are staggered end cutters. And the recognition sequence could also vary for each of these enzymes. So for example, here, this is a very frequent cutter or HAE3 because it is a 4-base pair cutter. The recognition sequence is only 4 bases. In comparison, NOT1 is a relatively rare one because it has a 8-base recognition sequence, which means that unless these 8 are present in the sequence given, you will not be able to cut with NOT1. Restriction enzymes form a very important toolkit of biotechnology. They give you a handle to manipulate the DNA along with the other DNA manipulating enzymes that we had done in lecture 2. And they are very commonly used in recombinant DNA technology, southern blotting, sequencing, etc. So with that brief on restriction enzymes, let's get started with restriction mapping. And before that, let me also discuss what are the types of maps that you commonly find in biotechnology. So there are 2 basic maps in biotechnology. One is known as the genetic map, which is based on the percentage recombination that happens between 2 genes. And this is also basically an exception to Mendel's law of independent assortment. Here, the distance between genes is measured in percentage of recombination or in centimorgan units. So if the percentage of recombination between, let's say EC and VG gene is 11%, then the distance is said to be 11 centimorgans. This is not a very high resolution map because the number of genes present in the genome is just 1.5% of the total genome. So even if all genes are linked, you would still be able to map only around 1.5% of the genome. So this is not a very high resolution map. And then the other type of map that is commonly used, and which is the topic of discussion today, is what is known as a physical map. In the physical map, you calculate distances between two DNA points in exact number of base pairs. So here in this case, I show you a restriction map. This is a linear DNA on which we have marked positions of enzymes A, B, and C. And very clearly between A and C, you can see there is a 1 kb distance. So genome mapping is used to identify and record the location of genes or other features in the genome and the distances between genes or other features in the genome on a chromosome. And genome mapping played a historical role in starting the Human Genome Project. Some of the key terms, so what is a genome? The genome is one complete complement of genetic material in an organism. So then mapping, of course, is where you want to exactly place on the genome what is the position of gene A, gene B, or other specific features, like let's say position of repeat or position of promoter. You could have a physical map where the distance between two features in the map is given in terms of the number of base pairs that separate them. And then you could have a genetic map. Genetic map was the first form of map, and it was it is a low resolution map where distance between genes is calculated on the basis of the percentage recombination that happens between them. The two genes have to be linked in order to be mapped, because in that case, the percentage recombination is less than 50%. So let's now talk of restriction maps. So here is your vector, and on this vector, we're going to mark the positions of three enzymes, PST1, EcoR1, and HINT3. All distances on this map would be in terms of kilobases. So here you are. This is your starting point, which is also known as the zero, or the if you make a full circle, this is 11.3 kb in length. So this is both your 0 point or the starting point and the 11.3 kb point once you make the full circle. This is where PST1 is located, and we are going to mark other enzymes with respect to PST1 now, right? So here is your next enzyme. This is your EcoR1, which is 2.5 kb downstream of the PST1. Then you have HINT3, which is 4 kb downstream of PST1. And then, of course, you mark all the restriction sites for the respective enzymes. And finally, you get a map that is known as a restriction map, where all enzymes are marked with respect to 11.3 or 0 position for PST1. If you want to calculate the distance between EcoR1 and HINT3, you know this would be 4 minus 2.5 or 1.5 kb. Likewise, the distance here would be 6 minus 4 or 2 kb. Here it would be 3 kb, and so on and so forth. So that is the whole idea of restriction map. And the question is, how do you arrive at a restriction map? And to arrive at a restriction map, you're given a combination of digestion products, and then you have to find the permutation and combination that satisfies those digestion patterns. So we'll come to that in a moment, and that is what the lecture is mainly about. And we'll take a linear DNA example and show you how restriction maps are made based on the digestion patterns given to you. So a restriction map is a diagrammatic representation of relative positions of restriction enzyme sites on a DNA sequence. So this is a restriction map where three enzymes, PST1, EcoR1, and HINT3 are marked, and we have taken PST1 as our reference point. And we are marking sites with respect to the reference, such as a 0 position. How distant the enzyme sites are with respect to the 0 position is what is marked here. So the band pattern obtained on digestion of the DNA sample with a variety of restriction enzymes, both singly and in combination, helps to construct a restriction map. And we'll go through this in more details as we move along. The resultant fragments are separated by agarose electrophoresis, and their sizes in base pairs are determined by comparing them with the molecular weight markers. So I'm sure you're now familiar with agarose electrophoresis and how do you determine the size of bands. We had done it in the lecture on PCR. We'd also done it in the lecture on restriction mapping. So basically, you run a molecular weight marker alongside, and that allows you to determine the size of the band that you have obtained. Then the process of assembling a map is a trial and error process. So how do you arrive to this final map is basically by trial and error. You, based on the digestion pattern that you get, you keep trying the fragments in various combinations until you hit a pattern that matches exactly the banding pattern that you've obtained on your experiment. So the process of making a restriction map is complex, and mostly it is a trial and error process where you try and rearrange the fragments that you obtain on gel in your experiment to see if they satisfy the conditions that, if they satisfy the banding patterns you observed in your experiment. And I've already told you that the correct map is the one that accounts for all the banding pattern observed on gel, right? What is also important to mention is, as the number of restriction enzymes increase, the number of possible correct maps also increase. So there are more than one possible solutions to a given banding pattern that you observe on gel. So more than one solution to a restriction pattern observed on gel is possible. But as I mentioned, the complexity of the problem increases exponentially as the number of restriction sites increase. We'll go through all these points as we solve a question. I'll give you a simple question first, and then we'll make it a little more complex by adding an extra restriction enzyme. And that should give you the idea of how a restriction map is made based on the banding patterns observed on gel on digestion with single or combination of enzymes that you're looking for. So before going into the complexities, let's observe some of the basic rules in restriction mapping. So as you know, DNA could be linear or it could be circular. And depending on what is the nature of DNA, the number of fragments formed on complete digestion would be slightly different. So here in this case, I show you a linear DNA, and I show you a linear DNA with one restriction site. When you digest this, you would obviously get two fragments. So this is a smaller fragment, and you have a larger fragment here. This one is a linear DNA with two restriction sites. When you digest this completely, by complete digestion, I mean both the restriction sites will be digested. So here in this case, again, if you do a digestion, so you get now three fragments, 1, 2, and 3. And then I show you another linear DNA. So if you do a complete digestion of this DNA, you'll get 1, 2, 3, and 4 DNA fragments. So when you have one cut site, you get 2 DNA fragments. When you have two cut sites, you get 3 DNA fragments. When you have 3 cut sites, you have 4 DNA fragments, which means that in a linear DNA, if there are n restriction sites, you'll get n plus 1 number of fragments, right? So that's something that is important again. For linear DNA, if you have n restriction sites, you'll get n plus 1 fragments. And then now let's talk of circular DNAs. So here is a circular DNA, and now if you see, this one has one restriction site here, and when you digest this now, all you get is a single fragment. Likewise, if you have 2 restriction sites on the circular DNA and you digest them, you get 2 fragments. This is the first fragment, here is the second fragment. Likewise, if you have 3 restriction sites on the circular DNA and you digest it completely, then again you get 3 fragments. So 1 restriction site for circular DNA, 1 fragment, 2 restriction sites for circular DNA, 2 fragments, 3 restriction sites for circular DNA, 3 fragments, 1, 2, and 3. So the general rule here is, for circular DNA, if there are n restriction sites, there will be n number of fragments, right? Now the question is, how can these fragments be arranged? So here you again have a linear DNA which has 2 cut sites, and now you know if a linear DNA has n cut sites, it will have n plus 1 fragments. So here 2 cut sites means 3 fragments, and let's say these fragments are named as A, B, and C, right? So this is your 3 fragments that you can obtain. Now the question is, in how many different ways can you arrange these fragments here? So let's see that here. So here you are, possible number of arrangements. If this is the 5' end and this is the 3' end, you will have 6 possible arrangements here: ABC, ACB, BAC, BCA, CAB, and CBA. Right? So what's the formula that you can use to calculate how many possible number of arrangements you can have for a given number of fragments? So the formula here is x factorial. So the number of ways in which x fragments can be arranged is x factorial. X factorial essentially means if the number of fragments is 3, then it breaks down to 3 into 2 into 1. If the number of fragments, total number of fragments obtained was 4, it will be 4 into 3 into 2 into 1. If it is 5 into 4 into 3 into 2 into 1. So that's again very important. The number of ways in which x fragments can be arranged is x factorial, and x factorial essentially means x into x minus 1, x minus 2, x minus 3 until it comes down to 1. Right? So that's the whole idea of it. And x, how do you calculate x? X would be n+1 for linear DNA, because linear DNA in restriction site possesses an n+1 fragments, and it would be n only for the circular DNA. So let us now look at a question. If a linear DNA and a circular DNA have the same number of restriction sites, which of the two will have higher number of possible arrangements? So let's say we have a DNA and these are the number of restriction sites that are there. Now we'll first talk about the circular DNA and then we'll go to the linear DNA. So here is your circular DNA, and you know in circular DNA the number of fragments formed on complete digestion would be n, where n is the number of restriction sites. So also the number of possible arrangements will be n factorial. So here you are. So when the number of restriction sites is 1, the number of fragment is 1. When it is 2, it is 2. Likewise, 5, 5, 6, and 6. And the possible ways these sites can be arranged is given by the formula of n factorial. So when it is 1, there is only one possible way of arranging it, because the closed circular DNA is only going to open up at that particular one restriction site upon complete digestion. If it is 2, then again you get 2 into 1, which is 2. If it is 3, then the number of possible arrangements is 3 into 2 into 1, which is equivalent to 6. When it is 4, it becomes 24, or 4 into 3 into 2 into 1. When it is 5, it becomes 5 into 4 into 3 into 2 into 1, which is equivalent to 120, and 6, it becomes 720. Likewise, if your DNA is not circular but linear, then in that case, again, you know that the number of fragments obtained will be n+1, where n is the number of restriction sites, and the number of possible arrangements that can come out of n+1 fragments will be n+1 factorial. So here you are. So this is the formula here. So for, let's say we compare for 6 restriction sites, in circular DNA you have 6 fragments, and they can be arranged in 720 possible arrangements. In linear DNA, you'll have 7 possible fragments, and the number of ways these can be arranged is 5040. So if you look at this here very clearly, with a linear increase in the number of restriction sites, the number of possible arrangements increases exponentially, both for circular DNA as well as for linear DNA. So the take-home message here is that the complexity of the problem increases exponentially as the number of restriction sites increase linearly, right? So here I show you the sample restriction map for circular DNA, and this one here is a sample restriction map for a linear DNA. In linear DNA, it is easy. The 5' end is marked as 0, or the start of the map, and the 3' end is marked as the total length of the DNA that you're investigating. And then, of course, the restriction sites are marked here, and for convenience, I mark the restriction sites at the bottom, and the distance from the reference, which is from the 5' end, on the top here. So this distance is given in kilobases with respect to the reference or the 5' end here, right? So the marking is a little more complex in circular DNA, because in circular DNA there is no open end. So what you do is you take one of the restriction sites as your reference and mark it as 0, and also the same point is marked on a complete circle as the actual length of the DNA. So this, for instance, is an 11.3 kb circular DNA. So we mark this point as 0, or the start point for PST1, and also for our convenience, we put it as the end point at 11.3 again. So there is also one more concept that you need to understand, that is known as complete digestion and incomplete digestion. So here is a restriction map of a linear DNA, and you can see there are 2 cut sites here, one at 6 kb, one at 14 kb. The total length of the DNA is 22 kilobases. So complete digestion essentially means that all sites and all DNA molecules are digested. So let's say you have 100 DNA molecules here in your solution, and you're digesting for both B and C. So every DNA molecule of the 100 that you have will be digested at C and at B as well, right? So now let's look at the number of fragments that you obtain upon complete digestion of this DNA. So in this case, you'll get 3 fragments, and the size of the fragments would be, this is your first fragment, that is 6 kb, 6 minus 0. This is your second fragment, which is 14 minus 6, that is 8 kilobases. And then you have a third fragment that will be 22 minus 14, again 8 kilobases. This is the pattern you obtain on complete digestion of this DNA piece here. So now let's look at incomplete or partial digestion. So here is the same DNA molecule again. And what is partial or incomplete digestion? So not all sites in the DNA are digested, because you are giving less reaction time. There is just enough time to digest either this site or this site here. So of the 100 DNA molecules that you have, approximately 50 would have a cut on C, and another 50 would have a cut on B, right? And accordingly, you'll have different fragment sizes. So if the cut is on C alone and not on B, then in that case, the fragments you obtain are 6 kilobases, 6 minus 0. And then the other fragment that you obtain is 22 minus 6, that is 16 here, right? If the cut is happening only at B, not because you have given only B, but because you have given C plus B, but you're not giving enough time for the reaction to go through. So in that case, again, you will have two fragments. This is the first fragment for 5' end. You'll have one 14 kb fragment. And then, of course, the remaining, that will be your 8 kb fragment. So effectively, what you get is 4 bands here instead of the 3 bands that you get on the same DNA on complete digestion. So the number of bands obtained is more than obtained on complete digestion. So now let's look at a typical restriction mapping problem and also see how the data for this restriction mapping problem is generated. So here you are. This is a typical restriction mapping problem that will be given to you. So in this case here, a linear DNA was cut with the following enzymes. The names of enzymes are B and C. These are hypothetical enzymes. Based on the digestion pattern mentioned below, determine the restriction map for this piece of DNA, and all fragments are given in kilobases. So when you digest with B enzyme alone, you get 2 fragments of 14 kb and 8 kb. When you digest with C enzyme alone, you get 2 fragments of 16 and 6 kb. And then when you digest with B plus C together, then in that case, you get 3 fragments of 8, 8, and 6. So this question represents a typical restriction mapping problem where the observed banding patterns are given to you, and based on that, you're supposed to draw a restriction map that fits in these patterns obtained on restriction digestion. So the question now is, how do you actually get this data for restriction mapping problem? And as is indicated here, this is obtained by doing restriction digestion of your DNA in question by enzyme B alone and enzyme C alone. And then in one reaction, you add both B and C and do the reaction and see the banding pattern that is observed on agarose gel electrophoresis. So here is what you do here. First, you set up 3 digestion reactions. You add your buffer, you add your DNA, and you add your enzyme or enzymes, depending on whether you're digesting with single enzyme or a combination of enzymes. So here is a reaction setup for enzyme B. You add the buffer, you add the DNA, you add enzyme B, and this final volume of the reaction is 20 microliters. Likewise, you set up a reaction for enzyme C. So this is basically you're looking at the fragmentation pattern of your DNA of interest when it is digested with C alone. And likewise, you have another Eppendorf tube where you set up a reaction for B and C enzymes together and accordingly reduce the volume of water that you add to make up the volume to 20 microliters. Right? So this is your experimental setup. The samples are incubated at 37 degrees centigrade for 1 hour, and after incubation, you basically check the size of fragments obtained on each Eppendorf tube using agarose gel electrophoresis. So here is your agarose gel. The blue area indicates the position where the wells are located. And in these wells, you're going to load the samples. For example, here we're going to load the fragments obtained from digestion with B alone. Here we load samples obtained with digestion with C alone. The fragments obtained on double digestion with B plus C and complete digestion are loaded in this lane. And in the last lane, you're going to load your molecular weight marker or ladder. Molecular weight marker, as it resolves, will help us ascertain the size of the fragments that we obtain in B, C, and B plus C lane here. Right? This is your negative electrode. Here is a positive electrode. DNA being negatively charged will move towards the positive electrode, and that will allow for the resolution of the bands on the agarose gel based on size. So as the agarose gel electrophoresis proceeds, the bands get resolved like this here. Right? The molecular weight marker profile is provided by the company and tells us the size of each bands in this profile here. And now that you know the size of these bands here, you can compare them to the obtained bands here to ascertain their size. So B gives you 14 and 8, C gives you 16 and 6 kb fragments, and in B plus C lane, instead of 3 fragments, you have only 2 because there are 2 cut sites. You expected N plus 1 for linear DNA, but instead you have only 2 fragments, but one of the 2 fragments is much brighter and thicker, which means there are 2 different fragments of the same size that are co-migrating to this position at 8 kb. So this one here is 8, 8, and 6 kb fragments, right? So let's now do a real question and see how you are able to make a restriction map finally from the observed band sizes that you get on digestion by restriction enzymes. So here is a simple question for you. The question is, a eukaryotic DNA was cut with the following enzymes. You have these enzymes B and C, and what you're given is the binding pattern obtained on complete digestion with B alone, on complete digestion of the DNA fragment with C alone, and the combined double digestion fragment pattern for B and C together, right? So this is what you have. Based on the following data, determine the restriction map, and you're given that all distances here are in kilobases, right? So now based on this, we have to draw a restriction map for this particular banding pattern observed on agarose gel electrophoresis. So the first thing that you need to decide is whether your DNA is linear or it is circular. So here in this case, it is clearly mentioned as eukaryotic DNA, and unless explicitly mentioned that you're referring to mitochondrial DNA or chloroplast DNA, the DNA can be taken by default to be linear because this is coming from nucleus. So eukaryotic DNA essentially would mean it is linear DNA, unless of course it is mentioned that it is coming from mitochondria or chloroplast. Likewise, if you write a prokaryotic DNA, it is by default considered to be circular. If you write plasmid DNA, it is by default considered to be circular again. So the information on the nature of DNA, whether it is circular or linear, can be explicit or it could be implicit, and you should be able to decide for yourself whether your DNA is linear or circular, because I've already shown you that for the same number of restriction sites, the number of fragments obtained in circular DNA is different from the number of fragments obtained in linear DNA. So now we know in this case here very clearly that our DNA is a linear DNA. So here you are, calculate the number of restriction sites to be plotted. For linear DNA, you know, n restriction sites give you n plus 1 fragments. So here in this case, on complete digestion with B, you get 2 fragments, which is 14 and 8 kb. So which means n plus 1 equals to 2 or n equals to 1. So there is one cut site for B that is to be marked on the map. Likewise, for C here, there are 2 fragments that are formed, 16 kb and 6 kb, which means n plus 1 equals to 2, which means n equals to 1 again. So total number of sites that you need to mark on the map finally is 2. And of course, when there are 2 restriction sites on the map and you are digesting with both the restriction enzymes, then in that case you get 3 fragments, which is as is shown here, right? So then B has 2 fragments, which means there is 1 cut site for B. C has 2 fragments, which means there is 1 cut site for C. Total sites to be marked with the map is equal to 2, right? And the number of possible ways in which this can be arranged, so if there are 2 restriction sites marked on a linear DNA and you have a complete digestion, then in that case, the number of fragments formed will be 2 plus 1, that is 3, and the number of possible ways it can be arranged will be n plus 1 factorial or 2 plus 1 factorial, which is 3 factorial, which is equivalent to 3 into 2 into 1 equals to 6. So the number of possible ways in which it can be arranged is 6. Now the challenge for you is to find that particular combination that satisfies these banding patterns observed on agarose gel, right? So first in the map, we mark the position of B. So we take B as our reference, and we know B on complete single digestion gives you 2 fragments, 14 kb and 8 kb. So the number of possible ways in which it can be arranged equals to 2 factorial, which is equivalent to 2 into 1 equals to 2 here, right? The other important thing that you need to do is to calculate the total length of the DNA, and that is very simple. You can take any of the single digestion patterns and sum it up together to give the total length of the DNA. So B, for example, on single digestion gives you 14 kb and 8 kb fragments, which means that the total length of the DNA is 22 kilobases. Likewise, C, for example, can give you 2 fragments, 16 and 6, which is again 22 kilobases. Or for that matter, if you take B plus C together and do a complete digestion, you get 8, 8 and 6, which is 8 plus 8, 16 plus 6, 22 kilobase pairs is the total length of the DNA. So the total length of the DNA is 22 kilobase pairs, right? So now we start plotting our map taking B as reference, and we know B gives you 2 fragments on complete digestion. One is a 14 kb fragment, the other is an 8 kb fragment. So the number of possible ways in which the fragments can be arranged is 2 factorial, which is equal to 2 into 1 equals to 2. So very clearly, either the 14 kb fragment comes first or the 8 kb fragment comes first. So to make the map, we refer to the top strand of the DNA and do the labeling on the top strand. So here is the top strand of the DNA. This is the 5' end, this is the 3' end. And you can see the 5' end is marked as 0 or the starting point, and the 3' end is marked as the total length of the DNA that you have in hand. So in this case, this is a linear DNA of 22 kilobases. Right? There are two possible maps of B, and both are correct because in both cases, single addition with B gives you an 8 kb fragment and a 14 kb fragment. So we take this map where we have marked the position of B at 14 as a starting point, and now we look forward to marking the position of C on this map based on the banding pattern observed here. Right? So this is our map till now. We have marked the position of B at 14. And now we are looking to mark the position of enzyme C on this map here, which is a 22 kb map with B marked at 14 kilobase pairs. So coming back here now, what we do is we now keep the two digestion patterns together. So we're looking at B alone, which gives us 14 and 8. And then we're looking at B plus C together, which gives you three fragments of 8 kb, 8 kb, and 6 kb. So very clearly, this 8 kb fragment of B remains intact on digestion with C. And actually, the 14 kb fragment can be summed up as 8 and 6 kb fragments. So this implies directly that C is within the 14 kb fragment of B, right? So you know that now the position of C is within this fragment and C is not in the other fragment, which is the 8 kb fragment of B. So C is now limited, or C, the position of C is fixed within the 14 kb fragment of B. Right? So C is within the 14 kb fragment of B and breaks into 8 and 6 kb fragments. So there are two possible positions at which now C can be placed on the 14 kb fragment of B. Either the 8 kb fragment comes first or the 6 kb fragment comes first. So let's place it together here. So here are two possible maps for C for a given fixed position of B. We have already fixed B at 14. So we're now looking at the position of C. And based on this banding pattern where the 14 kb fragment of B breaks into 8 and 6 kb fragment on double digestion with C, you could have the 8 kb fragment first, or you could have the 6 kb fragment first, right? So these are two possible positions, and now the challenge is to identify which of these two positions is correct. So now to check which of these two maps is correct, we look at the C single digest here, right? So the C single digest gives you 16 kb and 6 kb fragments. So if you look here, when you do a single digestion with C here, you'll get an 8 kb fragment and a 14 kb fragment. Likewise, when you do a single digestion with C here, you'll get one 6 kb fragment, which corresponds to what you observe here, and another 16 kb fragment, which corresponds to, again, to what you actually observe in the banding pattern. So in that sense, now you know that this is not a map that is possible. The actual correct map is this one here. So C alone gives you 8 and 14 kb fragment, which is not the case in your observed banding pattern. In this case here, C alone gives you 6 kb and 16 kb fragment, which matches to the observed banding pattern for C in the question. So which means now that this map is correct and this map is incorrect. So to check for which of these two is correct, we cross-check for single digestion of C, which as per question should give you 16 6 kb fragments, which are observed here but not here. So therefore, this map is correct for B plus C, while this map is incorrect. Right? So here is our final map for two enzymes BC and the observed banding pattern. So you have B at 14, C at 6. There are only two cut sites to be marked here. So we have marked two restriction sites B and C. And this map now satisfies all the conditions mentioned in your question. And let's now do a little more complex question than this. So let's add one more restriction sites to this and see how the complexity increases and how much more we have to work to get the restriction map. So here you are, and this is a new question now. A eukaryotic DNA was cut with the following enzymes. Based on the following data, determine the restriction map. All fragments are in kilobases. Here we have an additional restriction site, that is A. And when A cuts into this linear DNA, as the DNA is eukaryotic, we take it as linear. It brings you three fragments, 11, 6, and 5, right? And then B and C are the same as earlier. So B gives you 14 and 8 kb fragments. C gives you 16 and 6 kb fragments. A plus B gives you 8, 6, 5, and 3 kb fragments. So this is again a double digestion now with A plus B, which gives you 8, 6, 5, and 3 kb fragments. A plus C gives you 11, 5, 5, and 1 kb fragments. And B plus C, as earlier, gives you 8, 8, and 6 kb fragments. So here in this question, if you see, B and C are the same. And now we have added one new restriction enzyme, that is A. A on complete digestion of the DNA gives you 3 fragments, 11, 6, and 5, which means n plus 1 equals to 3, which means the number of sites that are present for A in this DNA are 2, right? So as in the previous question, we start first with calculating how many sites are to be marked, right? So here you are, calculate the number of restriction sites to be plotted. For linear DNA, n sites equals to n plus 1 fragments. So this 3 is n plus 1 for A. Therefore, n equals to 2, or the number of restriction sites for A equals to 2, right? So here you are, A giving you 3 fragments, which means there are 2 sites for A. B gives you 2 fragments, which means n plus 1 equals to 2, or there is 1 site for B. C gives you 2 fragments again, which means n plus 1 equals to 2, or n equals to 1. So total sites to be marked on the map now are 4 in number, right? And all these enzymes have to be marked on the restriction map in such a way that they represent the observed a single complete digestion by single enzymes or combination of enzymes here, right? So let's get started again. And this time, this is a little more complex than the previous question because the number of sites have increased. So the number of ways these can be arranged would also increase. And if the total sites on a linear DNA are 4, the possible number of fragments are 5. And therefore, the possible number of arrangements is 5 factorial, which is 5 into 4 into 3 into 2 into 1, which is equivalent to 120. So of the 120 possible arrangements, we have to look for the arrangement that satisfies our condition. And also, what is important to remember is, and I pointed out earlier also, is the fact that you could have more than one possible map for the same combination of patterns that you see, right? So again, our challenge in restriction mapping here is that of the 120 possible combinations, which ones satisfy the given conditions here, right? So let's get started again. We have to mark 4 restriction sites here, and we already have the positions of B and C, so we can take that as reference and then start marking for A. So total sites to be marked as 4, taking B as reference. We say because B has only 2 fragments, there are only 2 possible ways in which B can be arranged. So this is 2 factorial equivalent to 2. For B, either the 14 kb fragment comes first at the 5' end or the 8 kb fragment comes first at the 5' end. Accordingly, we have 2 maps here, and we take B at 14 as our reference and take this forward as we've done in previous question. So till now, we are exactly doing what we done in the previous question, right? So we take B at 14, we mark our restriction map as starting as 0. The end is marked as 22 kb because the total length of the DNA remains the same. If you see A, 11 plus 6, 17 plus 5 equals to 22 kb. For B single digest, 14 plus 8 equals to 22 kb. C single digest, 16 plus 6 equals to 22 kb. A plus B, 8 plus 6, 14, plus 5, 19, and 3, 22. Right. A plus C, 11 plus 5 is 16, 16 plus 5 is 21, and plus 1, 22. And B plus C, you know already, 8, 8, and 6. So this is 16 plus 6, it is 22 kb again. So let's now move on and try and see how we fix the position of C here. And this is again as per the previous question. So you now compare the B digestion alone with B plus C double digestion. And when you see that here very clearly, the 14 kb fragment of B breaks into 8 plus 6 kb fragments. The other 8 kb fragment remains intact here, right? So now you know that C is within the 14 kb fragment of B and breaks it into 8 and 6 kb fragments. So now you can place C at 2 possible positions. And as we had done last time here, so either the 8 kb fragment comes first within the 14 kb fragment of B, or the 6 kb fragment of C comes first within the 14 kb fragment of B. So these are 2 possible positions for C. One is where the 8 kb fragment of B plus C double digestion comes first. So you see here, and then you have the other one where the 6 kb fragment of B plus C double digestion comes first. When you do the final, you cross-check this map with looking for single digestion of C. In this case, when you digest single with C, this gives you a 8 kb fragment and a 14 kb fragment. which is not what you observe when you do a single digestion with C. So which means that now, because this single digestion C condition is not met, this map is incorrect. Now you move on to the second map here, and then again you check for the single digest of C. And here in this case, you get a 6 kb fragment, and then 22 minus 6, which is remaining 16 kb fragment, which means then that this map is correct. Right? So for B plus C, we have already fixed the positions. Next challenge is to fix the position of A now, right? So let's now look at how do you go about fixing the position of A in this map, given that you have already fixed the positions of B and C. So we take this map forward and look for the positions of A in this mapping question. So here you are again, and until now our map is this. We start with 5' end and we mark positions on this map until the 3' end 22 kilobases. We mark the starting position as 0 for our convenience. So now we move on to try and mark A. And for marking A again, we take B as our reference. B alone gives you 14 and 8, and B plus 8 together give you 8, 6, 5, and 3. So if you see, the 8 kb fragment of B remains intact on double digestion with A, which means all the A cut sites are within the 14 kb fragment of B. Right? So this is very clear now that all the A cut sites are here in this region, not in the 8 kb fragment of B. Right? 6 plus 5 is 11, 11 plus 3 is 14. So this 14 kb fragment of B splits into 6, 5, and 3 on double digestion with B plus A. So which is again, the implication is that both the cut sites of A are within the 14 kb fragment of B, because the 8 kb fragment of B remains intact and remains the same size on digestion with B plus 8 together. So the 8 kb fragments remains intact and is not digested on double digestion with A plus B, which means that both A cut sites are within the 14 kb fragment of B, and break it into 6, 5, and 3 kb fragments. Then we also take into reference A plus C now. So A plus C, if you check, it gives you 11, 5, 5, and 1 kb fragment, right? So now we want to place the first A site here. So what we'll do is we'll place the A sites with respect to 6, 5, and 3 kb fragments that we get from B, and cross-check whether A plus C gives you 11, 5, 5, and 1 kb fragments when you place these restriction sites on the 14 kb fragment of B, right? So here you are. So of these, again, you can check if the 6 kb fragment could be the first fragment, 5 kb fragment could be the first fragment, or 3 kb fragment could be the first fragment. So let's do it in the order it is given here. So let's place the 6 kb fragment which we get from B plus A double digestion first. So we place the 6 kb fragment first and on the 14 kb fragment of B. So we place A at 6. We already know that C is also at 6. So therefore, when you do a double digestion with A plus C, you should be getting a 6 kb fragment. So you check with what you get for C, and there is no 6 kb fragment here. So there is no need to further check this. You know that this position is not correct, right? So if the 6 kb fragment of A was first, then both A and C will be at 6, in which case A plus C double digest will give you one fragment of 6 kb, which is not the case here, which means that A cannot be at 6 kb within the 14 kb fragment of B, right? So this map is incorrect. So now let's put up the 3 kb fragment first and see whether it satisfies the A plus C double digest condition or not. Remember, restriction mapping is a process of trial and error. So here you are, and you're looking at now placing the 3 kb fragment of B plus A double digest first, which means that we're placing A at 3 kb in the map, right? So here you are, A at 3 kb in the map. You already have positions of C and B fixed at 6 kb and 14 kb. So now we place the 3 kb fragment first, and now we check for A plus C double digest. Remember, A plus C double digest that is given here is the final A plus C double digest where both A cut sites are present. So here in this case, when you place A at 3, you get one fragment that is 3 kb, you get a second fragment that is 3 kb, and then the remaining would have been 22 minus 6, that would be 16 kb, right? So some of the addition should give you 16 kb, and at least one of the fragments should match to 3 kb. So if the 3 kb or if the 3 kb fragment of B plus A double digest is coming first, Then on A plus C double digestion, you should get at least one fragment that is 3 kb. Remember, we have not put up the second site yet, so we are anyways not going to get all the fragments here, but at least one fragment would be 3 kb here, which is not the case here. The double digest for A plus C gives you 11, 5, 5, and 1. So therefore, this map is also ruled out. Right. So now we try for the last thing where we hadn't tried the 5 kb fragment first. So let's put that up and see whether it partially matches with the A plus C double digestion, because we're only putting at the first A site and not the second A site. So here you are. So now if the 5 kb fragment is first, A plus C double digest will give you a 5 kb fragment and a 1 kb fragment. And this is what matches to what we actually get here. So when A is placed at 5 kb, you see if you do an A plus C double digestion, you'll get a 5 kb fragment, and between 5 and 6 you will get a 1 kb fragment. So this is what you get here, a 5 kb fragment and a 1 kb fragment. Remember, we haven't kept the second A cut site yet. So if this were to match to this map here, it should actually be representing the 16 kb fragment. So 6 to 22 is a 16 kb fragment, and if you see 11 plus 5 gives you 16 kb fragment. So which means now that this position of A is definitely correct for the first cut site of A. So therefore, now you know that this first position of A, which you have marked within the 14 kb fragment of B by placing the 5 kb fragment of double digest of A plus B first, is the correct map till now. So you are now fixed with the first position of A. Now we look for the second cut site for A in this map. So here we are. Until now, what we have done is we have placed the B cut site, we have placed the C cut site. This was relatively easy because each had only one fragment and you could very easily place them. For A, we had to do a bit of trial and error and see which fragment comes first within the 14 kb fragment of B. And we had the 3 fragments, and we found out that when you place A at the 5 kb position, you satisfy partially the double digest obtained on A plus C. So we are placing A with reference to B and checking whether the position is correct for the final map with reference to C. And you see you have 5 kb fragment and you have a 1 kb fragment, which are clearly represented here as 5 and 1. And the remaining fragments where you have not placed the second cut site of A, you have a 16 kb fragment, which is 11 plus 5, which is equivalent to 16 here. Right? So this is the map here. So now that you've marked the first A cut site, you are left with marking only the second A cut site, and that will finish your map. And of course, again, there are 2 possible fragments which can come next to the 5 kb fragment of A. So it could be a 3 kb fragment or it could be a 6 kb fragment. If the 3 kb fragment comes next, which means that 5 plus 3, the next A cut site would be at 8 kb. If the 6 kb fragment comes first, which means that 5 plus 6, which is 11 kb, and the next fragment of A, next cut site for A would be at 11 kb, right? So therefore, now we make both the maps and see which one is correct. So let us now try by placing the 3 kb fragment next to the first A cut site, which means that A would be 5 plus 3 at 8 kb on this map. So what I now want you to do is to place the 3 kb fragment of A next to the 5 kb fragment, which means if it is 5 here, the next 3 kb fragment of A would be at 8 kb in the map here, right? And see whether the A plus C double digest condition is satisfied or not. So I give you 30 seconds for doing that. And then of course, we'll discuss how this goes on. So here is your time now, right? 6, 7, 8, 9, 10, 11, 12, 13, 14. All right, so here are your 30 seconds, and I hope you all would have been able to map it and see that this is not the correct map. So let's see what happens here. So here you are. You have now placed the 3 kb fragment, which is obtained on digestion of A plus B together, which means that A is at 5, which is the initial position of A, which I found to be correct. The next 3 kb fragment is here. So A is at 8 kb in the map here, right? So now if you check for double digestion with C plus A, and now remember, you have also marked all the restriction sites. So now you should have a complete match with respect to C plus A. So if this is our correct map, let us see what we get on double digestion with A plus C. So here you are, this is the first cut site, so you get a 5 kb fragment. Then between A and C, you get a 1 kb fragment. Then between C and A, you get a 2 kb fragment, and then you get the remaining 14 kb fragment. So the double digestion pattern that you obtain if this map is correct is 5, 1, 2, and 40. So this is a 5 kb fragment, this is a 1 kb fragment, this is a 2 kb fragment, and this is a remaining 14 kb fragment. We are talking only of A and C double digest, so we are ignoring B here, right? So therefore, what you get is C plus A double digest gives you 5, 1, 2, and 14, which is not a match for what you actually get in your observations. What you get in your observations is 11, 5, 5, and 1. So which means again that this map is not conforming to the observations made on gel. So therefore, this is the incorrect map. Now we move on and we now place the 6 kb fragment immediately after the 5 kb fragment and check whether this satisfies our conditions for all the observations that we've made here. So here again, we move on and we now place the second A cut site 6 kb downstream of the first cut site. So 6 kb downstream means if the first cut site of A was at 5 kb, the next cut site would be at 11 kb. So here you are, this is 6 kb distance. So the second cut site of A is now at 11 kb, and now again we check for whether this digestion satisfies the observed binding pattern for C plus A double digest. So, and what I want you to do is to now calculate for yourself what fragments you get on digestion with C plus A double digest. So here we are, we're only looking at A and C together. So this is your first fragment, which is 5 kb. And then between 6 and 5, you get a 1 kb fragment. Then between the C site and the next A site, you have 6 and 11. So 11 minus 6 gives you 5 kb fragment. And then between A and the final fragment, the terminal fragment, we're ignoring B because we're looking only at A plus C double digest. So B is not to be taken into consideration. So between this and this, it is 22 minus 11, that is 11 kb. So what you get is 5, 1, 5, and 11 kb fragments. So here, if you match now, so 11 is 11 here, 5 is 5, 5, another 5 is 5 again, and then you have 1 matches here. So which means now that this map is the actual correct map. So congratulations, you have arrived at the right map here. This is a map that satisfies the position for B, for C, and the positions for A. And we can do a double cross check again, right? So let's do a cross check with all the whether this map satisfies all the conditions or not. Of course, we've derived in such a manner that we kept satisfying the previous conditions. So it would satisfy, but it is always good to cross check again. So here you are, this is the final map, and we first check for, let's say we check for single digest with A. So A gives you 11, 6, and 5. So these are the positions of A here. So A is 1 is at 5 kb. So 5 minus 0 is 5 kb fragment. 11 minus 5 is your 6 kb fragment. This one is 6. And then 22 minus 11 is your 11 kb fragment. So it satisfies all the conditions for A. Let's check at A plus C double digest. So A plus C double digest gives you 11, 5, 5, and 1. So this is your 5 kb fragment here. Between 5 and 0, this is your 5 kb fragment. Between 6 and 5, which is the digest coming from A and C, is your 1 kb fragment. So this is a 1 kb fragment. Then from this C site to the next A site, there is a 5 kb fragment. 11 minus 6 is 5. And then from this A cut site to the terminal end, you have the 22 minus 11 or the 11 kb fragment. So A plus C is also satisfied. Now let's look at A plus B, which we have not checked until now. So A plus B, if you look here, gives you 8, 6, 5, and 3. So here again, A plus B, this is your A cut site here. So you have a 5 kb fragment. Check this one. 5 to 11, you have a 6 kb fragment. Check this one. 11 to 14, you have a 3 kb fragment. Checked. And 22 to 14, 8 kb fragment. Right? So everything that you have in your board is satisfied now. We have satisfied for A, B, and C. We have placed in such a manner they satisfied the conditions. And B plus C double digest. Also, if you see here in this map, B plus C is what you're looking at. So ignore A. This is your 6 kb fragment. Then ignore A again. This is your 8 kb fragment between 14 and 6. And then again, between 22 and 14, it is another 8 kb fragment. So this is 8, 8, and 6, right? So now, very clearly, all the conditions are satisfied, whatever has been observed in the banding pattern. So this is the correct map. So the correct restriction map will satisfy all band patterns of the non-gel, whether it is a single digest with A alone or B alone or C alone, or it is a combined digest with two different enzymes. So this is a double digest of A plus B, A plus C, and B plus C. So this is our correct map here, right? So as I told you earlier, there can be more than one possible solution to a restriction mapping problem. So now, since that you have already derived the first map, the question is, can you derive an alternate map using this map as your reference? So here you are, you basically again draw a map, and this time I'm showing in a large view here. So I mark my 5' index 0 and the 3' index 22. And on this map, I will start marking positions of these restriction sites in the reverse order on the first map here. So if you see here between B and the terminal length, the distance is 8 kilobases, 22 minus 40. So that is our first position on this new map here. So we mark B at 8 kb here, right? Now we take B as reference here and look for the next restriction site in this map. So this is at 11 and this is A. So this is 14 minus 3, that is 11. So which means here it will be 8 plus 3, that is 11 again. So at 11 kb, you'll have A site here, right? So this is your 3 kb here, and you have an A site at 11 kb here. So now we'll look at the position of the next cut site here, which is C, and it is 5 kb downstream of A in this map. So now from this A cut site until the next cut site, which is at C, the distance is 5 kilobases. So which means from A, 5 kb downstream, you can mark another site, and this site would be your, for the cut site would be for C here. So we take this here and we mark C at 16. Next, if you see between C and A, there is a 1 kb gap. So you can put one restriction site at 17, and that should be your final map here. So this is your final map.